Okay, so let's get started. Good evening, good morning, everyone. So welcome to this time Immune Zoom seminar. So today is my great pleasure to welcome our today's speaker, Yichuan Xiao from Shanghai Institute of Nutrition and Health, Chinese Academy of Science. So I know Yichuan for quite a while. Just to give a very brief introduction of Yichuan. So Yichuan actually got his bachelor degree from Hubei University, Wuhan. And then he continued his study in cell biology in also Shanghai Institute for Biological Science, Institute of Health Science. And then he got his PhD on 2010, from where he continued to pursue his career and then to do a postdoc in MD Anderson in Houston, which he's become really productive during the postdoc period. Which afterwards, from 2015, he went back to China and then stayed in his original institute to become his own laboratory. And then he started his own independent career. So Yichuan actually did a really fantastic PhD program and then also postdoc training in T-cell biology, particular focus on the signaling, T-cell signaling pathway related to the tumor immunology. And then when he now after he become independent, his research interested focus on the T-cell agent, which is actually a really leading question in the T-cell biology field, and then focus on the aging process related to the immune regulation. I think during his independent period to now, he's really continued his productivity as his postdoc period, and really published a lot of influential manuscript, particular, again, focus on the T-cell aging related to the autoimmune and the inflammation. So without further ado, I would like to give Yichuan's platform. Please go ahead, Yichuan.
Okay, thanks. Thanks, Trans. Introduction, and good morning and good evening with friends in China and in the United States. And I'm Yichuan Xiao from Chinese Academy of Science. And my topic today is KU-mediated DNA sensing in T cell potential aging-related autoimmunity. As we all know, the world is becoming an aged world. According to the data from the World Health Organization, the age distribution in 2010, it looks like this very same pyramid. And when it comes to 2050, it was estimated that this pyramid will become much fatter, which means that there will be more and more aged person. And in China, it will be around 30% aged person when it comes to 2050, which means, and this will definitely increase the medical burden of the whole society, and aging is becoming a global challenge. During the physiology aging process, the immune system, immune organs such as thymus and spleens are the first to undergo senescence. And this year, also there's a paper published in Nature which suggests that an aged immune system can drive the senescence and aging of solid organ. And this is also maybe the reason that aged persons are prone to develop several autoimmune disease. So for our scientists, and I think it's quite urgent for us to study the aging, what drives the immune cell aging, and how does the immune aging induce senses or pathology of the solid organs. And the T cell is known to be the major effector cell to induce the autoimmune disease, and thymus is the major organ to produce the naive T cells. With the age increase, the thymus is gradually shrink, which are defective to produce the naive T cells. However, many studies have suggested that in the aged individuals, including human, mouse, and the peripheral T cells number largely maintained, and which is comparable with the peripheral T cells in the young humans and mouse. And this is maybe because of the aberrant T cell homeostatic proliferation, and which also contribute to the aging-related autoimmunity. However, what's the mechanism to drive this aberrant T cell homeostatic proliferation is not known. And on the molecular levels, because of age, can induce the DNA damage, which can cause a lot of free DNA in the circulation system, and this DNA was phagocytosed by a lot of immune cells in the immune cells, and this phagocytosis DNA was recognized by the well-known DNA sensor CGAS, then induce immune activation. However, when the T cell have this process, it's not known, and in addition, the loss of function mutation of this DNA degradation enzymes, such as the TREX1 or DNase1, and it was associated with several human autoimmune diseases in the mice. And also TREX1 knockout can cause severe autoimmunity and even the mortality. And if we delete the T cells, the autoimmunity and mortality was compromised, which suggests T cell may also play a role in the DNA sensing induced autoimmunity. And then our question is that how does DNA can sense the DNA? How does T cell sense DNA, and whether the T cell mediated DNA sensing they can contribute to aging-related autoimmunity? To address this question, we firstly connected the young and aged mice and the young and aged human T cells, and we found that there are more accumulated DNA in the cytoplasm of the aged T cells from either isolated from the mouse or the human CD4 T cells, and we can see here in the aged CD4 T cells, we also observe that there has mild DNA damage, which suggests by the gamma H2AX staining. And we also examine the endogenous DNA accumulation in the cytoplasm with by qPCR, which also suggests in the aged CD4 T cell there are a lot of endogenous DNA fragment like L1 and the TERT expression in the aged CD4 T cell, and also the accumulated DNA was positively correlated with the gamma H2AX levels. So which suggests this mild DNA damage may increase the cytoplasmic DNA accumulation. Then we also found that in the aged CD4 T cell, the p53, which is a DNA damage response protein, was constantly activated either with or without the anti-CD3 plus CD28 stimulation. And if the p53 was knocked out and the cytoplasmic DNA accumulation was greatly enhanced. Then we also use several DNA damage inducer to induce mild DNA damage. We can see here, if we expose the T cell with very short-term ultraviolet or treated with very low dose of CPT1, they cannot induce the cell apoptosis. If we expose the UV with high, very long time, like 180 seconds or or treated with the CPT1 with 1 micromole, they can induce a huge apoptosis. However, with this low-dose DNA damage-induced treatment, we also can observe a lot of cytoplasmic DNA accumulation, which suggests that the mild DNA indeed can mediate the cytoplasmic DNA accumulation in the CD4 T cells. And then we asked what's the consequence of this DNA accumulation in the CD4 T cell. We found that T cell stimulation will greatly enhance the T cell proliferation after transfect with this double-strand DNA named ISD. And also in the TREX1 knockout T cell, which will cause the endogenous DNA accumulation. Also, we observed that the TREX1 knockout, they can also greatly promote the T cell proliferation. But if we transfect the DNA-1 to degrade the cytoplasmic DNA, and the proliferation will greatly suppress and abolish the difference between wild type and the TREX1 knockout T cells. And also the similar phenomenon was also observed in the young and age-derived CD4 T cells, and we found that age CD4 T cells show the increase in damage. They can increase the DNA accumulation. They can respond more actively to the T cell stimulation, show the much more increased the DNA T cell proliferation. And if we transfect the DNA SD or DNA-1 to degrade DNA, and the cellular proliferation was greatly enhanced or decreased with no difference between young and age CD4 T cells. And moreover, when we treat the CD4 T cell with a very low dose of DNA damage inducer with CPT1 or ultraviolet exposure with very short times, you can see here that this low dose CPT1 treatment or short time UV exposure, they can also induce greatly enhance the T cell induced cellular proliferation, and also p53 knockout, they can also promote the cellular proliferation of CD4 T cells. And similarly, if we degrade the DNA with DNA-1 in p53 wild type knockout CD4 T cell, and their proliferation was greatly suppressed and with no difference between the two groups. This data connectly suggests that mild DNA damage indeed they can promote the DNA accumulation in cytoplasm and then promote the CD4 T cell proliferation. And what's the in vivo function? And then we found that the DNA accumulation also promotes the T cell induced interferon gamma production. However, the interferon beta production, which is known that in the immune cell, DNA sensing can induce type 1 interferon signal activation. However, in the T cell, we found the interferon beta production was not affected either transfecting with a vehicle or double-strand DNA, and which suggests that the DNA sensing T cell maybe not active with the type 1 interferon signaling. And when we compare the young and aged CD4 T cell, we also observed that the activation related gene expression like IL-2 and interferon gamma was also greatly enhanced after the T cell stimulation in the aged CD4 T cells. And also in the TREX1 knockout mice, and the interferon gamma would also increase upon T cell stimulation. And then we found that when we transfer the aged CD4 T cell or the young CD4 T cell that's transfected with DNA, they can have enhanced ability to induce the autoimmune inflammation in the regular knockout mice. Then our last question is, what is the specific DNA sensor in CD4 T cells? So we perform a mass spectrometry screening after pull down of the biotinylated the DNA that we transfect in the in the ZAK T cell, we found the top two hits are Ku70 and Ku80, which are two proteins of the Ku complex. And we also found that DMPK, which is direct downstream kinase of Ku complex, was also co-IP'd with this Ku70 and Ku80. The the co-IP analysis also confirmed that when we pull down the biotinylated DNA and the Ku70, Ku80 DMPK can also be co-IP'd with this with these proteins. And however, the CGAS, which is a well-known DNA sensor in the immune cells and other cells, cannot be pulled down. And we also didn't detect the CGAS by mass spectrum screening. And we also found that in the Ku complex protein, including Ku70 and Ku80, were abundantly expressed in the cytoplasm of the CD4 T cells. And we also confirmed by the immunofluorescence analysis, which suggests that the Ku70 and the double-strand DNA were co-localized in the cytoplasm of the CD4 T cells. And to confirm that CGAS, whether CGAS whether or not CGAS involved in the DNA sensing in CD4 T cells, we treated the wild type and the CGAS knockout or STING knockout of the T cells. We transferred with the cells with the double-strand DNA. We found that T cell stimulation does not affect the cellular proliferation either CGAS knockout or STING knockout. And moreover, when we pull down the CGAS or Ku70 in the primary CD4 T cells, the transfected with the double-strand DNA, or we found that only Ku70 can efficiently co-IP this DNA, and also Ku70 showed a much more higher efficiency than to pull down the endogenous DNA fragment. And the CGAMP activity assay was also confirmed that in the CD4 T cell, we cannot detect any CGAS activity. However, in the macrophages, we can detect there are much higher CGAS activity as indicated by the CGAMP levels. So this is maybe the reason that the Ku70 and Ku80 was highly expressed in the adaptive immune cells like CD4 T cells, A T cell, and B cell. However, in macrophages, they have much more CGAS expression, and with very low levels of the CGAS protein in these adaptive immune cells. It's also confirmed by the immunofluorescence analysis. So therefore, in CD4 T cells, the DNA was sensed by the Ku complex, but not the CGAS STING system. So our next question is that how does this Ku-mediated sensing activate CD4 positive T cells? Then we performed our sequence analysis of the aged and young CD4 T cells. That upon TCR stimulation, we found that TCR stimulation we found that the T cell activation related signaling and pathway were greatly enriched in the aged CD4 T cells. And type 1 interferon, you can see here, except the interferon gamma, the other type 1 interferon related gene expression was very low, suggests the T cell cannot be active this signal pathway. And the KG analysis suggests that the AKT signaling was the most enriched signaling pathway in the CD4 T cells when compared with the young CD4 T cells. And we also the GSE analysis also confirmed that the enrichment of AKT signaling, and we also found that enriched several autoimmune diseases like the systemic sclerosis and lupus autoimmunity. And then we found that in the CD4 T cells, T-cell stimulation, they can greatly enhance the DMPK phosphorylation, which is the direct downstream kinase of cool complex. And we also confirmed that the AKT and the downstream S6 activation was also enhanced in the CD4 T cell, or the CD4 T cell, they transfect with DNA, or in the TREX1 knockout T cells. TREX1 knockout T cells. If the TREX1 knockout T cell was degraded with the DNA was degraded, and this DMPK, AKT, and S6 activation were greatly suppressed in the TREX1 knockout cells. So this data confirmed that the sensing, they can activate the AKT and AKT mTOR signaling pathway, and furthermore, to confirm this data, we use a small molecule inhibitor which named STL1127705, which is a inhibitor. They can interrupt the association with cool complex and DNA, and you can see here the immune block the analysis confirmed that this inhibitor treatment, they can greatly they can abolish the DNA binding with the cool complex, and functionally, if we treat this inhibitor STL, you can see here they can greatly suppress the DMPK activation and downstream AKT and mTOR signaling, either in the T cell that transfect with DNA, or in the CD4 T cells, or the CD4 T cell that transfected with the T cell derived self DNA. And similarly, if we knock down the Q70 and Q80, or we knock down DMPK, and the downstream AKT and mTOR signaling will also greatly abolished in the human junkie T cells. So this data will further confirm that DNA sensing indeed mediate the AKT mTOR signal activation in the CD4 T cells. Okay, so next we check the in vivo function, and if we treat the you can see here the STL, this inhibitor treatment also suppress the T cell active the proliferation in the CD4 T cells, and either in the CD4 T cell or the T cell transfected with double-strand DNA, and this inhibitor, and according to this inhibitor, they can greatly suppress the autoimmune inflammation in the aged older mice, in the aged mice. So our last question is, how does this pool-mediated sensing activate AKT mTOR signaling in CD4 T cells? So we firstly perform the in vitro kinase assay. You can see here that the PS0K could efficiently phosphorylate AKT, directly phosphorylate AKT in vivo, and however the DMPK, they fail to phosphorylate AKT, the fullness AKT in vitro, which suggests that the DMPK is not the direct kinase to activate AKT. So we perform the mass spectrometry screening again, and to search for the cool complex downstream kinase that mediate the AKT activation. You can see here the DMPK was the top one hit, and the second one is the CDK11B, which is a kinase that localized in the nuclear. So we focus on the our study on the third one, and then ZAK, which is a kinase that localized in the cytoplasm, and the co-IP analysis confirmed that ZAK, they can bind with Q70 and the CoA-T, and also they can bind with AKT when stimulate with the anti-CD3 plus CD28, and this kinase expression would also enhance in the CD4 T cells or the CD4 T cells stimulated with the anti-CD3 plus CD28, and the in vitro kinase further confirmed that DMPK they can directly phosphorate the ZAK at the screening side, and ZAK they can directly phosphorate AKT, and if we co-transfer with DMPK with ZAK, and this DMPK they can further promote the ZAK-induced AKT phosphorylation, and if we knock down the DMPK, the gene name is PRKDC, and the ZAK-induced induced AKT phosphorylation was greatly suppressed, which suggests that ZAK is a downstream kinase, direct kinase DMPK to mediate the AKT activation in the desensing pathway in CD4 T cells. And similarly, we also performed on sequence analysis. We found that in the ZAK wild-type CD4 T cells, we also enriched the AKT signaling when compared with ZAK knockout T cells, and the GSEA analysis also phenocopy the signaling and the disease enrichment in the aged CD4 T cells when compared with the young CD4 T cell. We also enriched the AKT signaling, cytoplasmic response, and JAK signaling, and systemic sclerosis and lupus autoimmunity. And if we knock out ZAK, we can see here that DNA-induced AKT signaling, AKT and S6 signal activation was greatly suppressed, and we also treat the wild-type ZAK knockout mice with stimulate with anti-3 and plus CD28, and then treated the cells with different AKT and upstream AKT upstream kinase inhibitors. We can see here that only AKT inhibitor and the DMPK inhibitor, they can abolish the difference between wild-type and knockout CD4 T cell, which suggests that AKT is the downstream kinase DMPK to activate AKT. And moreover, we found that in the p53 knockout, if we further knock out ZAK, they cannot affect the DNA combination in the cytoplasm of the p53 knockout mice, and then they cannot affect the DMPK activation. However, they can greatly suppress the p53 knockout induced the cellular proliferation, and similarly we can find that they can knock out, they can greatly suppress the DNA transfection induced the cellular proliferation, and in vivo we found that in the wild-type aged mice, the autoimmune inflammation was greatly enhanced. However, in the ZAK T cell conditioned knockout mice, and this aged CD4 T aged mice cannot further be enhanced the autoimmunity. And next, we performed the mass spectrum again to identify the activation site of ZAK. We found that actually we identified several phosphorylation sites, and finally confirmed that threonine 169 is the activation site of the ZAK, and we also we then generate this phosphorylation antibody against the threonine 169 of ZAK. And then we found that TREX1 knockout indeed promote the TCR-induced the ZAK phosphorylation at this site, and if the DNase1 can greatly suppress its activation, and similarly the double-strand DNA transfection also promote the ZAK phosphorylation at this threonine site, and also STL which is the inhibitor to interrupt the cool complex association with DNA, also suppress the ZAK activation in response to T-cell stimulation. And then we generate a T169A knock-in mice, and we found that this screening site knock-in, they can greatly suppress the DNA-induced AKT and downstream AKT mTOR activation, and accordingly the T-cell proliferation was also greatly suppressed. And it is well known that the dieting can greatly improve the aging-mediated pathology. So we also tried the dieting model, including the calorie restriction or fast mimicking dieting and model to check whether the dieting can affect the DNA accumulation or DNA sensing in the CD4 T cell. And interestingly, we found that the dieting they can greatly suppress the DNA cytoplasmic DNA accumulation in the HCD4 T cells, and accordingly the ZAK phosphorylation and the threonine 169 and the AKT activation and the T cell proliferation was also greatly suppressed in the HCD4 T cell in the HCD4 T cell that isolated from mice that treated with dieting. And similarly, the dieting mice showed much lower autoimmune diseases. And then our final question is that, can we target this DNA sensing pathway to treat the aging-related autoimmunity? Then we collaborate with collaborate with Zheng Mingyue, who is a chemist. They based on the ZAK crystal structure, and they screen by using the artificial intelligence screening technique. They screen a library that contains around 130,000 compounds. Finally, we got a 114 candidate compound by using the virtual screening, and then we synthesize this 140 candidate compound and then perform the biochemical analysis to examine their inhibitor effect against the ZAK enzymatic activity. We found finally we got one compound named ZAK2. They can efficiently suppress the ZAK kinase activity, and this is the ZAK2 structure. And we found that this compound, they can bind with ZAK protein and several amino acid sites, and they can also efficiently suppress the ZAK activity with IC50 of around 1.5 micromole. And we also found this inhibitor can be selectively suppress the ZAK activity among the 85 Cas panels. And more interestingly, we found this compound, they can efficiently suppress the DNA-induced AKT and mTOR signaling pathway and the T cell proliferation either in mouse CD4 T cells and human CD4 T cells. And this inhibitor can also successfully suppress the autoimmune inflammation in the aged mice. So finally, this is our working model. So when the aging comes, and they are not all circulating free DNA, which can be effects of endocytosis by the T cells, and also the aging-induced DNA, they can promote the DNA breaks, and this and then this breaks DNA can be entered in the cytoplasm, and which cause a lot of DNA accumulation in the cytoplasm of the CD4 T cells. And this accumulated DNA was recognized by the Cu70 and Cu80, and this cool complex further recruit the DNPK. And the DNPK, they can activate the ZAK and the threonine 169 and then activate ZAK, they can further activate AKT and downstream mTOR signaling, which promote the T cell activation and proliferation related gene. And finally, to promote the autoimmune inflammation. And we also draw a cartoon to explain this mechanism, and which is this cartoon shows the ancient Chinese mystery story named "How Issues the Sun," and in the sky, in the ancient time, there are 10 suns in the sky, which represent the uncontrolled proliferation of T cells. If we target this sun with the DNA sensing inhibitor like ZAK2, we can shoot down the sun and we can relieve the autoimmune inflammation. And our paper was recently published in the Immunity, and in the same issue, the Immunity also recommend our paper with a preview, and which suggests in the immune cell like macrophage, they have very high level of CGAS and low level of cool complex, and the CGAS, they can recognize the body region of the DNA, and then they can activate STING and downstream TBK1 R3 to promote the type 1 interferon expression. However, in the CD4 T cells, that with the very low level of CGAS and high level of the cool and the cool complex, they can recognize the end of the DNA, and then to activate the downstream DNPK, ZAK, AKT, and mTOR signaling axis, and then promote T cell activation and proliferation. And I propose that and these two effects, and this effect, combined effect, they can promote the aging-induced DNA sensing and autoimmunity. So, and this work was mainly completed by my PhD student Wang Yan, and also get help in that member. And I also would like to thank our collaborator, Professor Zheng Mingyue, and their help with the small compound screening and synthesis, and also the biochemical analysis. And I also would like to thank our several collaborators who gave us a lot of help, including Professor Liang Yinmin, Zhong Bo, Huang Xinfang, Xiao Hui, Shu Hongbin, Li Hao, Ge Baoxue, Xu Pinong, and so on. And I also like to thank the fundings from the Chinese Academy of Science and National Science Foundation of China, and the most. And I also would like to thank our institute platform. And finally, thank you for listening, and I'll stop here for the questions. Thank you.
Thank you for Yichuan's great talk. I think we can start with some questions. I see Xiaolei has a question. Xiaolei, go ahead.
Hey, Yichuan, this is really nice work and elegant mechanism. So I have a couple of questions. So I think first, so I'm trying to understand like what's the physiological function of this double-strand DNA or exogenous DNA induced T cell proliferation. So why do CD4 T cells like react to this like DNA, and what do they do? Like, do you have any like comment?
Actually, I think that you know, different cells use different DNA sensing system. This is our, yeah, this is what I think after we completed this study. And you know, in the immune cells, they have a lot of CGAS, so they use the CGAS to recognize DNA. And in the CD4 T cells, they have they can use the cool complex, and they use they use different DNA sensors. They can activate different downstream signaling pathway. And actually, we also did some unpublished data. We have some unpublished data which suggests in some cells, they can both use both two systems, the cool complex mediated DNA sensing and the CGAS mediated DNA sensing. Actually, for example, in the CD8 T cells, they have some CGAS expression, and actually we have data show that in the side that in the adaptive immune cells, among the CD4 T cells, CD8 T cells, and B cells, actually the CD8 T cells have native high levels of CGAS. So, and this also paper published that indeed CD8 T cells, they can use CGAS system to produce interferon beta to against the tumors. And we also actually confirmed by our experiment that when we pull down the CGAS or co-sampling in the primary CD8 T cells, they can both kind of co-immunoprecipitate with the induced DNA. However, in the B cells, there has very low level of the CGAS, and in CD4 T cell also has very low level of CGAS, and only the co-sampling can efficiently pull down the DNA. And also, actually, we also test some other tumor cells, but not all the tumor cells. Actually, we found that in some tumor cells, they can definitely use this mediated sensing pathway to promote the tumor growth. And so I think there's a lot of things we need to to figure out in our future works. Yeah.
Okay. Yeah. Thanks for the additional introduction for the CD8. I think my question is more about, is this like co-mediate mechanism in CD4 T? Is that because CD4 is misled by these exogenous DNA to think that it needs to increase the immunity in response to pathogen infection, or there's some like physiological reason that why do they want to do this, even if it can cause some autoimmunity?
Yeah, for the T cell, actually, I introduced in the background that because this is not usually common thing in the young CD4 T cells, in the CD4 T cells from the young individuals, and when there are not of the endemic or circulating DNA under the aging condition, and in the age human or mouse, even that because of the Alzheimer's shrink, and they can cause the defective production and naive T cell. However, the peripheral T cell number was not maintained, and so they need a mechanism to maintain this the T cell number, which is called the homeostatic proliferation. So I think that this DNA-mediated T cell proliferation maybe is the major driver for the maintenance of peripheral T cells in the aged human and mouse, and maybe other mammals. So I think that's the major physiological function. Yeah,
that's and also more like a compensation in the cell membrane.
Yeah, yes, more like compensation, and
but also more in a non-specific manner rather than like a specific.
Yes, I think it's some kind of non-specific manner. However, actually, we also have some data, unsure data, suggest that because the upstream DMPK. The ZAK activation needs the DMPK to activate them, and this DMPK activation that also requires the TCR stimulation. And DMPK, they can after when the cool complex they recognize DNA, and the cool then recruit the DMPK, and DMPK they can auto-phosphorylate. And we also found that TCR stimulation can further enhance the DMPK phosphorylation. So we speculated that maybe there are some kinds of or unknown mechanism, they can promote the DNPK activation, you know. So, and however, if we stimulate the T cells with the PMA and myosin, it's kind of not so obvious actually. So this DNA synthesis pathway is kind of, you know, more sensitive to the TCR stimulation, but not the PMA in myosin stimulation. I didn't show the data, present data here, but actually we have the data, you know.
I see. So that's interesting. So that means it's probably acting more upstream, these like a calcium or PKC pathway. So basically it means more proximal to the TCR.
We expected that maybe there's also kinase to promote the DNPK activation. Actually,
That's very interesting. So I assume like the Ku complex expression level is higher in the aged CD4 than the young CD4, right?
Actually, Ku complex expression level was comparable, was not so obvious. Yeah.
I see. So basically, in the young CD4, it can also, if let's say we transfect some plasmid into the young CD4, it will also like induce this Ku-mediated cell proliferation.
Yes, yeah.
I see. So the machinery is already there. It's just because I think a related question I'm interested in is like if we just compare the young CD4 versus the aged CD4, is there any like difference general like either on the TCR signaling pathway or on the DNA repair pathway? Did you find any like difference, let's say just by RNA-seq or proteomics?
Actually, we also examined the other signal pathway, but we examined the mKB and MAPK, and also the upstream phosphor RCK, phosphor NAD. It kind of not too much difference. Either the transfect with the DNA, with or without DNA transfection, when stimulated with the TCR. So and then this is the reason why we focus on the AKT and tossing. It's kind of the most effective signal molecule, the AKT and the downstream S6. So
I see. So could I just say, like in general, like just on the components of these the T cell signaling pathway, there are not much difference between the young and aged CD4 T cells.
Basically.
I see. So that's great. So last I have maybe a technical question. So the Ku complex is like it's involved in the non-homologous end joining, so DNA repair. So it's mostly localized inside the nucleus.
Yes, so, that's true.
So how is that because it's also there a field in the cytoplasm that it can detect the cytoplasmic DNA?
Actually, that's a very good question. Yes, actually, we examined. We also examined. We have data. In the manuscript, that because the Ku complex Ku70, Ku80 together with the DNPK, it's a well-known mediate the DNA damage response in the nucleus. Actually, so when we found that when we found the immune bond, we found that this Ku complex Ku70, Ku80 was highly expressed in the cytoplasm, and when in the nucleus, when this Ku complex mediate the DNA damage response, they also need a ligase. The gene name is XRCC4, and interestingly, if we knock down the XRCC4 and this signaling, the cytoplasmic signaling activation, like the AKT mTOR signaling, was not affected, was intact, was not affected. Only, I have shown data that only we knock down the Ku70 or Ku80 or DNPK, the AKT and mTOR signal was affected. So we think, the cytoplasmic DNA sensing mediate AKT and mTOR signaling is it's independent of the nuclear DNA damage response, you know. So this is our data, yeah.
So there are basically two pools of.
Yes, I think two pools. Actually, in CD4 T cell, I think there are more. There are more cool compounds in the cytoplasm than in the nucleus. Yeah.
I see. So one last small question. I think in the initial images you show a staining of double-stranded DNA, which is like mostly in the cytoplasm in these aged CD4. So does that like? But I didn't see anything in the nucleus. So I'm trying to understand what like is it specific recognized?
Yeah, that's a good question. You know, in the nuclear, the DNA was how to say that, was in the chromosome. It's compact, you know,
Yeah,
by some
state. Okay.
histone protein. They cannot expose the, you know, they cannot be detected. Actually, this is kind of common method by using they have some, you know, specific anti double-strand DNA, which antibody, which were we were purchased from the APCAM company.
I see.
Actually.
Okay, that's great. Cool. Yeah, thanks so much. It's really inspiring work.
Thank you. Yeah, thank you. Okay.
Ni hao, I think you have a question.
Hi, Dr. Xiao. Very nice talk and very clear presentation. I really enjoy it. I just have a very quick question, and I want to know your thoughts about the specificity of CD4 cells. Based on one of your slides, if I remember it correctly, I found that Th17 differentiation specifically is also enhanced. But based on your whole talk, you are just talking about CD4. And based on this cool mediated activation mechanism, it seems to me that maybe this mechanism applies to all the CD4 activation in different conditions, including EAE. But I'm just curious about, have you tried other models like Th2 activation CD4 models? And then can you also comment about this activation in different type of CD4? For example, will that also activate Treg as well? So Treg may be also affected. But why in your pathway analysis, it's only Th17 activation found?
Yeah, thank you. Yeah, this is a good question. Actually, you know, the activation, proliferation, and the differentiation is kind of, you know, it's totally different. You know, I know that I also read the paper suggest that, you know, the Th17 is kind of enhanced in the CD4 T cells, and also there are much more Treg cells. We also actually examined the tested that. And confirmed data that Treg cells is also, you know, there are more Treg cells in the aged mice, and actually our mechanism is kind of you know, is kind of that more focused on the T cell activation and proliferation, and also in the, you know, in the aged mice, we also have some unpublished data suggests not all the T cell proliferation was enhanced in the aged CD4 T cells. And there are some T helper subset is indeed, you know, it's defective. It's very defective. Actually, it's defective. Actually, so not all the T cell is differentiation is enhanced, and actually, so our project is kind of more focused on the proliferation and activation, and except for the EAE model. Actually, EAE is not a very good model to test the aging-related autoimmunity. Actually, right now we are testing the, you know, the rheumatoid arthritis and the lupus autoimmunity. They also show the similar effect that, you know, if there are more DNA accumulation in the CD4 T cell, and the ability to induce this kind of autoimmunity is also enhanced. So for the differentiation, just what I said, I think it's kind of, you know, the different pathways. So Yeah.
For example, have you checked the type 2 immunity model, like dermatitis?
For the type 2 immunity, yeah, I didn't check the type 2 immunity. Yeah.
I see. Okay. Yeah, thank you.
Okay.
Yeah, it's very interesting.
Yeah.
Phenomenon.
Thank you. Yeah.
I think Xiaoqing has a question. Do you want to ask by yourself or?
Okay, Xiaoqing.
Okay.
Okay. Let me see the question. Do you think the increase in plasma DNA or CD4 T cell may come from? Yeah. This is a good question. Actually, we indeed tested whether this increased DNA is come from the nuclear leaking or the mitochondria. Actually, both contribute to actually we indeed tested both contributed either from the nuclear and the mitochondria, but the majority is kind of from the nuclear. And we also tested when we deplete the mitochondria or by using some inhibitor and it kind of the nuclear leaking DNA can contribute much more to this DNA-induced CD4 T cell proliferation. Yes, yeah, that's a good question. Yeah.
Okay, so I have a couple of questions. First of all, I think in your data you show B cell, CD8, CD4, and macrophage. Seems like B cell has similar CGAS or the double-strand DNA as the CD4. Is that means that the B cell has similar aging process in terms of DNA sensing as the CD4 T cells?
Actually, we don't know. We are trying to figure out, in the future. Yeah. We also, indeed, we found the B cell have very high levels of this kind of cool complex expression, and and they also have very less CGAS expression, and how B cell sense DNA is also not known actually in this field. So we don't know yet.
But then it seems like CD4 and CD8 has quite different components of the cool complex, and which means the CD8 has different response compared with CD4.
Yeah, it's different. Actually, according to our unpublished data, which suggests the CD8 they can definitely use a cool contact to sense DNA, and there's also have papers show that the CD8 T cell also use the CGAS to mediate the type 1 interferon production to against the tumor. Yeah. I think both two machines works in the CD8 T cells. Yeah.
So, but on the other hand, in terms of biological significance, if you say that the CD4 T cells, when they're aging, they are more susceptible to autoimmunity and then respond to proliferation. But if that's the case, so how about in different scenario, for example, during the infection, which that means the response from the CD4 will be more rapid from the aging T cells?
Yeah, that's a good question. Actually, the reviewer also asked our kind of similar question. They didn't ask the infection. Actually, we also examined the cytoplasmic DNA accumulation in different scenario. Like if we stimulated the CD4 T cell with the TCR, but we didn't check the infection condition, and it is known that TCR stimulation also can induce the mild DNA damage, and also we found that the TCR also could promote the cytoplasmic DNA accumulation in CD4 T cell. So our explanation is that the TCR they can promote they can induce a mild DNA damage in the CD4 T cell, and this increase the DNA and also further promote the TCR-induced the T cell activation and proliferation. It's kind of a positive feedback loop. So this is how, kind of our explanation. And like you said, I think that the infection may also have this kind of mechanism. Also they can promote the CD4 T cell, the DNA accumulation and activation. However, they also maybe have some, have some mechanism to antagonize its effect. So I think it is very complicated. It's not only one mechanism works. And it depends on which one is the dominant effect under its condition. Yeah.
But when you talk about antagonist, can you be more specific? Because I mean,
I don't know actually.
Okay. Because I mean, to me, it's curious that autoimmunity and then infection probably all related to TCR activation, but for sure that the antigen specificity determine the T cell response. So, but I'm not sure if the phenotype you described in your working model relate to the autoimmunity. It seems like it's more general issue, but if it's a more general issue, then it should have no how you distinguish between different scenarios. That's will be important to dissect. Otherwise, I mean, one hand it could increase the risk of the autoimmunity, but on the other hand, which means it also should have a better response to infection diseases, which will actually is not the case, because we know that aging will actually compromise the outcome of the infection. Okay, so
Yeah.
I think my last question is, when you talk about the dietary treatment, I didn't quite get what is the FMD?
Is fast mimicking dieting.
Fast. Okay, fast mimicking. And when you check the CD4 T cells, where these T cells come from? From the spleen or blood?
You mean the mouse T cells?
Yeah.
It's isolated from the spleen and the lymph node.
I see. So you didn't actually, so the dietary treatment, usually how long you actually give the treatment to the mice?
For the diet, actually for the, we have two models, and diet for the calorie restriction model, we, we cut the cut the calorie by by 30, around 25 to 30%. This is the commonly used that calorie restriction model. And the fast mimicking model is kind of we use kind of like let the mice starving or calorie restriction. I'm not so sure. Actually, I need to check the protocol for around 4 days or 3 days, and then let them eat freely for the rest of the day of the week, and then the 2 cycles of the fast mimicking dieting, and then we collect we sacrifice mice and then collect the T cells, to examine for the examination. Yeah.
I didn't quite remember on these dietary treatment, although you see the change of the DNA sensing phenotype, that's the, except the immunofluorescence staining. Did you also check the signaling downstream, like the Ku complex and the phosphate ZAK kinase?
Yeah, dieting. Yeah, you can see here we use the flow cytotomic analysis. You can see here.
Yeah, this is the outcome, like the T cell proliferation and double-strand DNA. My question: have you ever checked intracellular signal after the dietary treatment.
Intracellular signaling.
Like you know, AKT phosphorylation. Oh, no, sorry, not cake. The ZAK phosphorylation.
Yeah, here.
Oh,
this
I
is
see. Okay, sorry. Okay. Okay, so well, thanks a lot for the great talk. Anyone else has questions? Okay, if not, then we thank Yichuan one more time for the great talk, and then really a enlightenment for the aging T cells. I think it's we learned a lot today, and then hopefully I will see you next week for other immune talks. Thanks a lot, Yichuan.
Okay.